Okay, so good afternoon everyone. You are joining the UP NISMED webinars on K-12 Science. Today's webinar focuses on the second quarter topic, living things and their environment. We hope that the webinar will help you understand the storyline that connects the 5 modules and give you practical tips for effectively facilitating the learning activities in your own classrooms. My name is Celia Balbin, and I will be your host this afternoon together with John Reyes. Both of us are from the Information Science Group of UP NISMED. Okay, so nawala na naman. Nawawala yung ating mga pictures. Okay, so I'd like to show to everyone our agenda this afternoon. We have allotted 1 hour and 10 minutes for the presentations and 10 minutes for the Q&A session. I am happy to inform all of you that in today's webinar we will have not 1, not 2, but 3 resource persons. Let me introduce them in order of their appearance in the webinar. So let's see. Alright, so our first presenter is Michael Anthony Mantala. Mike is a graduate of BS Biology from UP Los Baños, and for joining NISMED's biology group in 2009, he taught biology for 10 years at the Philippine Science High School, Cagayan Valley campus, and in 2 private science high schools. Mike also worked with 2 review and tutorial centers here in Metro Manila, and in his capacity as tutor handled all science subjects. Today, Mike will walk us through the storyline connecting the 5 modules of Quarter 2 focus and will present the overviews for modules 1, 4, and 5. Okay, let's welcome our next presenter, Miss Maria Dulcelina Sebastian. Dulce is a graduate of BS Biology from the University of San Agustin in Iloilo and holds a master's degree in biology education from UP Diliman. Before joining NISMED in 1985, she taught biology at the high school and college levels. And as science education specialist here in the biology group, Dulce has been involved in curriculum development, teacher professional development, and research. In today's webinar, Dulce will guide us through Module 2, Plant and Animal Cells. And our third resource person is Dr. Risa Reyes. Risa obtained her biology qualifications from bachelor's to master's to PhD degrees from UP Diliman. Risa belongs to NISMED's elementary science group and has extensive experience as curriculum developer, trainer, and researcher. Risa is also currently NISMED's Deputy Director for Research and Extension. For her share of the webinar presentations, Risa will walk us through Module 3, Living Things Other Than Plants and Animals. Everyone, please get ready. We are about to start the presentations. Alright, so I am putting this is the first presentation to be given to us by Mike Mantala. Let me clear that camera pod and activate this share pod for Mike's picture. Alright. Okay. So there we are. To present the storyline and introductions for Module 1, let's all welcome Mike Mantala. Mike?
Thank you, Ma'am Celia. Good afternoon, dear participants. Thank you all for making it to this webinar. This is the second webinar in the K-12 series. Now, for this afternoon, we are going to tackle the whole of second quarter, living things and their environment. My role is to present the storyline behind the sequence of topics covered in the 5 modules for this quarter and to give an overview of the said modules. I will also be presenting Module 1 right after my presentation of the storyline, and also Modules 4 and 5 later in the webinar. Okay, so the storyline provides a transition from Quarter 1, that's diversity of materials in the environment, to Quarter 2. It weaves all the concepts, principles, and processes in the second quarter through the fabric of 3 organizational levels: organism, population, and ecosystem. The storyline also draws attention to some unifying themes that cut across the different disciplines of science: parts and functions, interactions, change and its dynamics, and diversity. So this is how the storyline, in terms of how it is woven through the organizational levels, would look like. The modules have been sequenced in a way that it radiates from the parts that make up an organism outward to the parts and processes that constitute populations and ecosystems. At the organism level, we introduce the students to the different parts that make up an organism and their functions. While this is not a new topic to the students, they have already been introduced to the different organ systems in grades 5, that would be for reproductive system, and 6 for the other organ systems. It is important that we emphasize interaction between and among these parts that make up bigger structures and the role that this interaction plays in helping one organism meet its basic needs and survive. Indeed, the whole story of biology is about organisms meeting their basic needs and survival. Sorry for that. At the population level, we introduce the students to reproduction, a process that takes place among individuals of the same species. There are 2 modes of reproduction: sexual and asexual. In this topic, we also should underscore perpetuation of species as the primordial function of reproduction. At the ecosystem level, we introduce the students to the different components of the environment, the interactions among these components, and how these interactions maintain a state of balance in ecosystems. Okay, now, this is how we map the contours of the storyline in terms of how the unifying themes figure in the organizational levels in each of the modules. Notice that Module 3, Organism Other Than Plants and Animals, which Dr. Risa will tackle in a while, cuts across all the organizational levels and touches on all the unifying themes. We'll have that later. Okay, so these are the standards you will find in the curriculum guide. They served as our guide in working out the modules and the teaching guides. Okay, so that's for the storyline. We proceed now to an overview of the modules. Okay, so This is now an overview of the 5 modules for Q2 or for the second quarter. They are now flashed on your screens. Okay, Module 1 deals with the different levels of organization in an organism. Note though that this is not your typical levels of biological organization, which start from the subatomic particles up to the level of the biosphere. We figured that to present the topic of levels of organization in that manner would defeat the purpose for which biology was designed in the K-12, that depth takes priority over breadth. Module 1 also deals with interactions between and among the parts that make up bigger structures. Emphasis is given on the idea that anything that happens to any of the parts that make up a structure affects that structure and eventually the whole organism. So that's for Module 1. Now for Module 2, this module zooms in on an important level of organization in an organism, the cell. It's the basic unit of life. It's a basic unit of structure and function in biology. In this module, the students will be introduced to the different parts that make up a cell. They will also discover that some parts are common to both plant and animal cells, while other parts are seen only in either the plant or the animal cell. Module 3 picks up from where Module 2 ends, that there are different kinds of cells. In this module, the students will be introduced to organisms other than plants and animals with focus on single-celled organisms. Whereas in Module 2, cells have been introduced as a part that makes up a structure, meaning where similarly structured cells are grouped, they form tissues. Here, cells are introduced as organisms themselves, capable of carrying out the different processes of life. In Module 4, the students are introduced to the process of reproduction. This is a kind of interaction that takes place at the population level or among organisms that belong to the same species. It also introduces the students to the 2 modes of reproduction, sexual and asexual. Emphasis is given to perpetuation of species, the singular most important reason for reproduction in organisms. And finally, Module 5 introduces the students to the biotic and abiotic components of the environment, their interactions with each other, and the changes that come with these interactions. Emphasis is also given on the levels of organization in an ecosystem, that is, population, communities, and ecosystem. You can revisit your discussion of levels of organization in an organism in Module 1 to transition to Module 5. Okay, so that's a brief overview of the modules. We now move on to the individual modules, starting with Module 1. Spiraling progression is one of the features of the K-12 curriculum. We've been reminded of that time and again. With this approach, topics that have been covered previously in the lower grades are revisited and used to deepen discussion in the succeeding topics. This is just a bite-sized spiraling progression of topics across 3 grade levels that defined Module 1. Perhaps what comes as striking to bio majors who have taught biology for eons in the old curriculum is not that new fancy concepts were introduced in biology in the K-12, but that fancy concepts were stricken out. This is to give way to concepts that are contextualized in real-life situations and are of real-life applications. For instance, in the old curriculum, the topic on levels of organization straddles quite a range from subatomic particles to the biosphere. Imagine that. What do the students make of that? Also, organ systems are taken up independently of the other organ systems. For example, the students will be taught the different parts that make up the digestive system and their functions. The students learn what happens to the food as it gets broken down to smaller pieces before it gets excreted out of the body. But what about the nutrients from the food that were assimilated and circulated to the different parts of the body? This gap is bridged by introducing the idea of organ systems, or parts for that matter, interacting with each other to help the organism meet its basic needs. With this in mind, we thought bio in the K-12 is bio just right. Let's look again at this slide. The topic of organization in an organism, the topic levels of organization in an organism starts from the macro or the readily observable, the organism, down to the micro or the not so readily observable, the cells. Okay, so these are the big ideas in Module 1 that we want our students to learn. That organisms are made up of parts that play specific functions, that these parts work together to help the organism meet its basic needs and survive, that anything that happens to a part affects the structure it makes up and the whole organism, and that we take care of these parts to keep them functioning properly. I hope we are able to make that clear with our students as we implement the modules and the teaching guides this quarter. In the teaching guide, you will find 8 items for the pre-test and post-test. This slide shows how each item is classified thematically. Of course, this is not hard and fast. This is not definitive. This is not the be-all and end-all that you can come up with. There are fine lines separating the themes from each other. Here now are the key questions for Module 1 and the activities that were incorporated. What are organisms? None of you students equate the term organism only to living things that move, like the animals. Those that do not are not organisms. They are plants, or something to that effect. What makes them up? This is a tricky question because parts are integrated according to levels. Again, none of you students would say organisms are made up of cells, blood, bones, stomach, eyes, and a muscular system. It is not wrong, neither is it accurate. Okay, so here are the objectives for Activity 1. Activity 1 is a very simple activity. However, as you notice in the teaching guide, it made use of a number of analogies and other representations. Caution was given as to its use because, done differently, it can result to misconceptions. To remedy this difficulty, it is proper that we define the limits of the analogies we used. Also, models are never, ever a substitute for the real thing. Where it can be helped, do the real thing hands-on. Here are the objectives for Activity 2. In this activity, students will be asked to interview relatives or neighbors who have had heart or kidney problems, of their experience coping with the disease. At times, interviews yield misleading information. What can be done is to compare this information with those they've read as they looked for pictures they will use in making a poster. They can get these pictures from newspapers, journals, or the internet. Okay, so I guess that's just about it for Module 1. I'm returning later for Modules 4 and 5. Mamsal?
For the storyline and also the Module 1 overview. Shortly, I will be turning you over to the next presentation, but let me take this few minutes to remind everyone. You can now start typing your questions in the chat window. We are going to collect them and we will later on answer those questions when we reach the Q&A session. So again, start typing in your questions. Alright, so right now I'm changing the presentation to that of Module 2. So please bear with me. In a while, I will be using Dulce's presentation. Alright, and also sharing her picture. So at least you have an idea who you're talking to. Okay, so we're ready. Here's Dulce for Module 2.
Okay, good afternoon, dear colleagues at KaSaMa. Let us now proceed with Module 2 of Quarter 2, entitled Plant and Animal Cells. It has been explained earlier by Mike that Module 2 also draws attention to some unifying themes of biology, and in this case, particularly parts and function, interactions, and diversity. So from the first module, the students learned that all organisms are made of cells. So the key questions for Module 2 are all cells the same? If not, in what ways are they different? What big ideas would our students learn after doing all the activities and participating in the discussions and as they go through the module? So the big ideas for Module 2 are all organisms are made up of cells with parts that work together for their survival. Plant and animal cells differ in shape and in some parts, and most cells can only be seen using the microscope. The student material for Module 2 consists of 2 sections. The first part is on cell parts with activities and discussion. The second part is on how to use the light microscope, also with activities and discussion. In the teaching guide, there is a pre-post test with 10 items consisting of multiple choice and open-ended types. It also contains 2 more additional items if activity 2 is performed. The pre-post test is also present in the section on how to use the light microscope. These pre-test and post-tests have accompanying answer keys. As for the learning activities, there are 2 for the cell parts section. If you notice in your screen, Activity 1 is on comparing plant and animal cells. Activity 2 is on investigating plant cells. In the section on how to use the light microscope, there are 3 procedures: procedures A, B, and C. And if you notice, there are asterisk marks in activity 2 and how to use the light microscope. This means they require the use of microscopes. Okay. For activity 1, on comparing plant and animal cells, the estimated time allotment is 1.5 hours, including discussion. The objectives in this activity, okay, are found in your screen. These are: the students should be able to identify parts of the cell, describe plant and animal cells, differentiate plant cells from animal cells, and construct a Venn diagram to show parts common to both and parts only found in either plant or animal cells. In this activity, the students will make use of 2 illustrations, the one for plant and animal cells. Okay, in the post-activity processing, please take note that when students present their Venn diagrams, they should look like either of the 2 shown on your screen. Other students may also make one in which the overlapping circles are lying one on top of the other. So as long as these elements are present, the ones that you see on your screen, then these should be accepted. Now we come to the part on how to use the light microscope. Please remember that in the student material, there is a reminder there that this should be done prior to activity 2. The suggested time element allotment rather, is from 2 to 3 hours. Procedure A will deal with the microscope, its parts and the function. Procedure B will be on making a wet mount, and procedure C will be on observing specimens. The objectives of the activity, there are 7 of them. So at the end of the activity, the students should be able to handle the microscope properly. They should already know the different parts of the microscope, describe what these parts are, and they should be able to prepare materials for the microscope study. The computation for the magnification of objects seen under the microscope is also included as an objective in this activity. Now, with regards the materials to be used in this activity, in procedure A, it is indicated that lens paper should only be used in cleaning lenses of the microscope. This can be bought in photoshops, but in case you cannot find them in your area, I think you're familiar with the cloth used in cleaning eyeglasses. This can also be used. Now, what are the error-prone steps in this activity? Please take note in procedure B that in steps 16 and 17, our students may make the mistake of not positioning the cover slip at a 45 degree angle against the slide and slowly lowering it onto the slide. Most of us know that this is to avoid trapping air in between the cover slip and the slide so that bubbles will not be formed. No, because if bubbles form, this will interfere with their observation of specimens. In step 22 of procedure C, students may also make the mistake of not looking from the side of the microscope when changing from the low power to the high power objective. This is to ensure that the lens of the high power objective will not hit either the cover slip or the slide and may cause breakage of these two, or even the breakage of the lens of the high power objective. Now, we should not also undervalue this part on the care and maintenance of the light microscope. Let us remember that we should give opportunity to at least more students to know about the microscope, its use, and more importantly, their opportunity to observe specimens or objects that cannot be seen by the unaided eye. At the same time, we would like also to teach our students responsibility of using equipment as they study the cells. I would like also to draw your attention to the corrections in the student module or student material. On page 96, figure 1 should read the light microscope and its parts. And on page 102, item number 6, in the second sentence, the silica get packs there should read silica gel packs. Let's move on to activity 2 on investigating plant cells. The suggested time allotment is from 2 to 3 hours, including discussion. And there you will find the objectives on your screen. There are 6 of them. Okay, so the students here should be able to prepare a wet mount. They should be able to describe the cell, the plant cell that they will observe under the light microscope. They're also required to stain plant cells and then observe the parts of the cell, identify the observable parts of the plant cell. They're also going to draw they're expected also to draw onion cells as seen through the light microscope, and of course to explain the role of microscopes in cell study. Okay, with regards to the materials for this activity, please use red onion. If iodine solution is not available in your laboratories, they are available in drugstores. If you don't have the scalpel, an ordinary knife may be used to cut the onion. And the forceps can be very well replaced with old tweezers that you are not using anymore. Just clean them with ethyl alcohol and then you can use them. For activity 2, with regards error-prone steps, okay, I would like to draw your attention to figure 3. Please take note that the correct way of preparing onion scale for microscopic study. Okay, so in figure 3 it is clearly shown how to do it. Kindly supervise your students while they'll do that. In step 2, let us ensure that students correctly place the onion specimen on the glass slide. In the student material, it is indicated that the transparent onion skin should be placed on the glass slide with the non-waxy side facing up. Please check also that the onion skin is not folded or wrinkled so that they can make good observations. In step 3, let us remind the students not to tilt the microscope while observing wet mounts, as we all know that the water from the wet mount may spill or wet the metal parts of the microscope and cause rusting. Step 4 in this activity would remind again students to raise the objectives a little and look to the side while changing objectives. No, the reason I have indicated earlier. And in step 6, please ensure that students exercise care in adding iodine solution to the specimen. They should avoid spilling the iodine solution on their hands and their clothes. Okay, and lastly, as teachers, please ensure that all materials and specimens to be used are ready prior to starting the activity. This would include availability of the materials to be used before the activity. And then please check that all microscopes are in good condition before you give them out to the students. And if you have important questions or issues that may not be clear to you, please don't hesitate. Always collaborate with and ask assistance from biology teachers in your school. So thank you very much. This is all for the second module. In case you have more questions or concerns, just email us. If you notice, our email addresses are right under our names in the first slide in our presentation.
Thank you very much, Dulce. Dulce has given us not just an overview of what students will learn in Module 2, but also given us some tips on how to deal with the materials used in the activities and some tips for avoiding certain error-prone steps in the different activities in the module. Alright, so let's move on. We're also taking note of your questions, the ones that you're typing in the chat window. After this presentation, we will move on to Module 3 presentation to be given to us by Dr. Risa. Again, I will be switching to her presentation right now. Again, thank you very much, Dulce.
You're welcome.
Just a moment, please. Okay, where the presentation is loading right now. Okay, so again, we will have Risa, so you will see her at least her picture.
Oh, that's me.
Okay,
See?
we're ready for you, Risa.
Okay, magandang hapon po sa inyong lahat, mga giliw na guro. I will walk you through Module 3, entitled Living Things Other Than Plants and Animals. Okay. So, here are the key questions and the big ideas. Yes, you will have a copy of this on this website, right, Celia? You will post it after our webinar.
Yes, that's right.
Okay.
They should
If you
see in the
recall,
announcement right under the...
Yep, the PowerPoint screen. Okay. So, in the early grades, in the elementary grades, the focus was on plants and animals. So here in grade 7, we will study the 3 other groups of the 5 accepted groups, widely known groups in biology. Okay? These 3 big groups have useful members as well as harmful ones. Like the other modules, this module 3 will also have a pre-post test. There are only 4 items. They are just examples, and you may add some more. And there's assessment for learning, meaning the activity sheets contains questions which are diagnostic as well as formative. That means you can monitor what your students are learning as they answer the questions and fill up the worksheets. So there are 3 activities. Are these also plants? What other living things are found in the school grounds? And the 3rd is optional. What do these living things look like under the microscope? I noticed one teacher said that she has 90 students, but only 1 microscope. So we made it optional because we know that there are schools which have no microscopes at all. Okay, these are the objectives of the first activity. Essentially, the teacher will show some examples of these 3 groups, which the pupils will observe using their magnifying lens, not the microscope, just a magnifying lens. Okay, like mushrooms, you'll see on your screen. Seaweeds, this is one kind. This is arosep or lato in the Visayas. Here are examples of green algae. This is another one, guso. Okay. And this is a lichen, which you know is a symbiosis of fungus and alga, green alga. Other examples to be shown by the teacher are a bacterial colony, because if you don't have a microscope, you can't view the individual bacterial cells. These are molds in a decomposing banana peeling. But you won't tell them that. You will see the questions in the activity sheets are asking pupils if they think these are plants and giving their reasons. So we'll just show examples first. Maybe they know about these, maybe they don't. That's what we'll find out when they answer the questions in the first activity. So this is a bread mold, right? As seen through the naked eye and using the magnifying lens. Baliktad ata yung aking pictures. This looks smaller anyway. And then lumot or green algae. If you notice in the Filipino language, lumot covers mosses as well as liverworts and green algae. That I've chosen to call our green algae lumot here. And you find them in bodies of water. If you're in the mountains, you'll find them in rocks or wet places. Okay. So, module 3 activity 1 will take an hour, one period, and the hands-on activity, observation, and answering the questions will take most of the time, after which for the teacher will facilitate discussion of their answers. And if you don't have seaweeds, you can use lumot, no? So, but the danger is you might, and even the pupils later on in the second activity, when they collect, they might mistake liverworts and moss for lumot because they are found in the same habitat. Of course, we have to listen to students' answers and their comments during the processing and discussion. That's what we highlight in our teaching learning materials. So in activity 2, these are the objectives. Essentially, you will bring out the students to the school grounds to collect specimens similar to the ones you showed. And they will bring this back into the classroom and draw them, describe them, and answer questions written there. Okay, so when you go out, you may see mushrooms like this one. Some growing on tree trunks. And lumot on rocks. And these are lichens, which may be found on tree trunks also, barks of trees, right? Different kinds, different colors, or even moist walls and rocks. So, since the whole module covers or is estimated to run for 4 sessions, activity 2 by itself may be done in 1 day. The hands-on activity, going out collecting specimens, then answering the questions, and then again, towards the end, the teacher will conduct a discussion of the students' answers. So the same error-prone step, no, mistaking liverworts and mosses for lumot. And as we have recommended, please listen to your pupils' answers to the questions in the activity. Okay. So for activity 3, which is optional, if you have a microscope, you will prepare slides as instructed in Module 2 and view sections of the lichens, bread molds, lumot, and the bacterial colony shown to the students in activity 1. So you will observe, the students will observe these living things under the microscope, draw and describe them, and label parts based on diagrams you show, like this one. This is the lichen, so it's a combination, symbiosis of algal cells and fungal hyphae. Like this one, this is a diagram, and this is a mount of a bread mold under the microscope, LPO, so the hyphae are evident. And under the HPO, they will see the spores, the sporangium, and even the stalk. Like this one too. And there's the diagram. You can ask them to label their drawings when they see this. So, this is the wet mount of a banana peeling, the molds. And lumot, you will see the individual cells there with their green parts containing the chlorophyll. Closer. There. The cells spoken about by Miss Dulce earlier. And if you have azolla in a pond in the school grounds, you could tease one leaflet. And under the microscope, you'll find Anabina azolla as cyanobacteria, which you have a slide here on your screen. Okay, I guess, okay, the microscope activity will also take. So, just some tips.
Go ahead, Risa.
Okay, go ahead, okay. Of course, when you take them out, you have to scout the school grounds ahead of time. That's advanced preparation, and identify the areas where each group will go. You should know which specimens they can collect there, and you can suggest this to them. Of course, they may bring into the classroom other living things that you didn't expect them to bring. Okay? Excuse me. You can find examples of the 3 other groups, fungi, protists, and monerans or bacteria. You can show slides or pictures, okay? So, I think that's it. Thank you very much. Salamat po.
Okay, thank you, Risa, for the Module 3 presentation. We are seeing a lot of questions now coming in through the chat window. Again, please note that we're collecting them for the Q&A session later after all the presentations. Okay, and I'm also seeing everyone, some people are asking if they can get a copy of the presentations. If you look a little bit below the presentation screens, you will find two rectangular announcements there, or notepads. One is titled Agenda, and the other one is labeled Announcement. In the announcement part, the one on the right, you would see that we have given you this bit of information that we will be posting in the KaSaMa Teachers online community site, the full recording of this webinar, all the PowerPoint presentations that are used here, as well as the list of all the questions that you've asked and the concerns and issues you raised during the webinar. So I hope, I am sure that that would be very helpful for everyone, especially the full recording and the PowerPoint presentation copies. Okay, so we will be moving on now to the next module, and that's modules 4 and 5, and we will be going back to Mike's presentation earlier, because as I have introduced him to you, he is the one who's going to provide you a discussion of these last 2 modules. So again, let me share Mike's presentation, and thank you, Dr. Risa. Thank you for doing module 3 with us.
You're welcome. It was a pleasure.
Okay, so I'm putting up Mike's presentation now. Okay, so maybe you can type in the chat window. If you can clearly see Mike's presentation on your screens.
Okay, thank you, Ma'am Celia. Again, good afternoon to our dear participants. I'm back for modules 4 and 5. Okay, so shown on your screen is a spiraling progression of topics across 3 grade levels that defined module 4. Okay, the topic of reproduction in the old curriculum is always introduced as a function of the of the reproductive system, and in most cases is always associated with animals only. In Module 3, the students should have learned that there are organisms that need not have tissues, organs, or organ systems to reproduce. To address this difficulty in the K-12, reproduction is introduced as a process through which organisms perpetuate their kind. There are two modes, sexual and asexual. Different organisms reproduce through different means, and this comes as a consequence of the diverse nature of living things. The concept of reproduction as presented in Module 4 does not involve hormones, feedback mechanisms, and the like that usually come with the topic. This is a calibrated omission. It would be pointless to be mentioning hormones, feedback mechanisms, and other physiological processes in reproduction given that the students have not been introduced yet to the major groups of biomolecules. Also, there is no association yet between sexual reproduction and recombination of traits or asexual reproduction and equational division typical of mitosis. Again, this is a calibrated omission. All these concepts are better introduced and concretized in the higher grade levels when the concept of cell division cycle has already been introduced. Okay, we take a look again at this slide. Module 4, as you notice, cuts across several unifying themes: diversity, interactions, and change. And of course, in Activity 3, the module will also touch on parts and functions, specifically the reproductive parts of a gumamela flower. Okay, shown here are the big ideas that we want our students to leave the classroom with. Firstly, reproduction is either sexual or asexual. Sexual reproduction involves sex cells. Asexual reproduction does not. Organisms produce more of their kind through reproduction. And lastly, knowledge of the different asexual means through which plants may be propagated finds application in agriculture. There are 7 items in the pre-test and post-test for Module 4, and they are grouped thematically as such. Okay, so shown here are the key questions for Module 4. What are the different modes of reproduction? How can we use this knowledge in growing plants? There are 3 activities that the students will do to help answer these key questions. In Activity 1, can you grow new plants from ice? The students will learn that tubers can be used to grow new plants. By the way, that's ice in quotation marks. This is showcasing to them some practical uses of knowing how plants reproduce asexually. In Activity 2, can one become two? The students will be introduced to other methods of asexual reproduction like budding in yeasts and fission in Protococcus. And in Activity 3, structure of a gumamela flower, the students will be introduced to sexual reproduction in plants using a perfect flower, meaning a flower that has male and female parts like the gumamela. Okay, so I think that's just about it for Module 4, Reproduction. Again, it's a very simple module with very simple activities. We move now to the last module, Module 5 for Quarter 2. Okay, so Module 5, the last module in this quarter, deals with the parts that make up an ecosystem, the interactions that take place between and among these parts, and the changes that result from these interactions. Shown in this slide is a spiraling progression of topics across 3 grade levels that defined Module 5. In this module, the students will be introduced again to the different levels of organization, except that this time, it will cover those that are beyond the level of the organism: population, community, and ecosystem levels. Note that the levels of organization in an organism have already been tackled in Module 1. You can use this to transition to Module 5. Like in Module 1, this is not your typical levels of biological organization, and just a segment of which is presented here almost on a piecemeal basis for the same reasons. We want to strike a good balance between depth and breadth according to the cognitive level demands appropriate for grade 7. We do not want to inundate them with information, leaving them flooded cluelessly. Okay, so let's take now a look again at this slide. Module 5 levels up the discussion on levels of organization started in Module 1. On interactions started also in level 1 and beefed up in Modules 3 and 4. And on diversity started in Module 2 and beefed up in Module 3. It provides the denouement. In literature, denouement is the unfolding of the plot towards the climactic part of the story. So with this module here, Module 5 provides the denouement in the storyline. The big ideas in Module 5 that we want to inculcate in our students are that, number one, the environment is made up of the biotic and abiotic components, that every part of the ecosystem is interrelated to all the other parts that make it up, and that these interactions, unless disturbed, keeps the ecosystem in a state of balance, and that through all these interactions, energy is transferred across trophic levels and from object to object, and is transformed from one form to another. Okay, so here are the key questions for Module 5. How do organisms interact with each other? How is... By the way, there are 8 items in the pretest and post-test that are classified again thematically as shown in this slide. Okay, moving on. Here are the key questions for Module 5. How do organisms interact with each other? How is energy transferred from one organism to the other? The students will work out 4 activities to help them answer these key questions. In Activity 1, what does it mean to be alive? The students will probe the things they see around them and decide on which properties are common among living things and which among non-living things. In Activity 2, Housemates, Ecomates. The students will set up mini aquaria and observe the interaction among the fishes, snails, and plants, and between these organisms and their environment. Note that oftentimes, students equate the term interaction only with organisms that move. For stationary organisms like plants, these students have only a vague idea as to how these organisms interact with their immediate environment. In Activity 3, which is what? The students will be introduced to the concept of feeding relationships and energy transfer. This activity should also be able to beef up whatever initial conceptions they already have about interaction. And lastly, in Activity 4, what to do with food wastes, the students should help, or rather, this activity should be able to introduce the students to the concept of cycling of materials through the process of decomposition, and that it should also be able to help students better appreciate the dynamics they encounter each day as they interact with their environment, and come to conclude that change is a brute fact of life that is at times good and welcome. Momsel, I think that's just about it. That's my presentation for Modules 4 and 5. Thank you.
Okay, maraming salamat ulit, Mike, for these last 2 module presentations. We have so far listed up quite a number of questions, and we are just right on time. We have very good pacing. It's a minute after 5 o'clock now, so we are ready to start with our Q&A session at this point. Okay, so open officially the Q&A session with a few questions here on our list. Let's see. Please allow me to start with the questions that directly relate to the presentations this afternoon. I will be skipping just now, even if they were entered ahead, questions that relate to assessment and the grading system. Alright, so and of course there are also again questions about when we are going to receive the copy of the students module. Until now, pag hindi pa po ninyo natatanggap yung mga modules ninyo, especially for the first 2 quarters, meron nga yata po kayong problema, because as Dr. Tan has told us earlier, the division offices have been provided the budget as well as the task of reproducing these materials for distribution to the different schools. So, pakikulit po yung inyong school's division superintendent if you still do not get copies of the student modules and teaching guides for the first and the second grading periods. Alright, so yun na lang po yung pinakasagot natin doon sa mga nagtatanong kung kailan pa nila makukuha yung kanilang mga modules. With regards modules for the third and fourth quarter, again, we were told by Dr. Tan in the last webinar that DepEd is saying that these modules and teaching guides will be made available in September, at least for the third quarter topic. Yung iba po, hindi po natin alam kung ano yung calendar ng DepEd, no? But I'm sure dapat po dumating sa inyo yon bago magumpisa yung third grading period at saka yung fourth or last grading period para naman dun sa Earth Science modules and teaching guides. Okay, so ngayon, let me check some of the questions here. There's one question from the Dasmariñas science teachers. We badly need the materials for the given activity. Earlier, I asked in the chat pad, ano pong activity ang sinasabi po dito? Or is this also referring to the modules that are not available yet, no? So, kung yun po ang ibig niyong sabihin, ay nasagot na po natin yun, no? If you're referring to the modules when you said the materials for the given activity, ay yun na po yung pinakasagot. Now, if you are very specific about a particular module and activity and which specific materials, pakitype lang din po sa ating chat pad so that our presenters will be able to give you the answer that you need. Without that detail, we will not be able to provide you a perspective into your question. Okay, so there's another one here referring now to Module 1. I think this question goes to Mike from the Dasmariñas science teachers. Some questions in activity in Module 1 is redundant. So it takes time for us to finish the Module 1. So, again, sana po na i-type po ninyo kung ano yung mga question numbers man lang po doon sa activity na nire-refer ninyo. So, if you can type that in later, we can go back to this question.
Okay, Marcel, as regards that question, that redundancy is actually a calibrated redundancy, meaning to say it's intended to scaffold the thought process of the students into what we actually want to achieve in that activity, and that is to answer the key questions. Now, yeah, it would take a while to finish that module, but I think it can be done. I think it can be done, and the redundancy part there, I think, has got to do with some questions in what they call this, in the presentation for animals being repeated in the presentation for plants. I think it's a calibrated redundancy, Mansell.
Okay, so again, there's another question still from Dasmariñas science teachers. Do we need to prepare the lesson exemplar, or we are just going to follow the suggested sample lesson plan by the DepEd Secretary Luistro? I'm not sure if we are familiar, if all of us are familiar with these sample lesson plans suggested by the DepEd Secretary. The question is, do we need to prepare the lesson exemplar, or are we just going to follow the suggested sample lesson plan? Do we have information about these sample lesson plans, Mike, Dulce, Risa?
Ma'am Sel, I think
Referring to.
Yes,
Yes,
Risa?
Mike, go on.
yeah, Ma'am Sel, I think Ma'am Jean has mentioned something about a special memo or an order from the Department of Education. There's a number there, I think memo number 70. I have not read the memo yet, and I know nothing about it.
Okay, actually, I've earlier downloaded that page showing the DepEd order. So if you allow me, I think this is dated August 13. So this is very, very new. Those of you who have the bandwidth still to connect to the DepEd website, you can see this DepEd order, no? But right now, it simply mentions that teachers' guides have been prepared for teachers, and there is a set of guidelines on the preparation of the daily lessons called the DLs. So if you want, I can show you this screen. Are you interested to see this DepEd order now, folks? Okay, so here's a comment from one of our participants. The lesson log is already in the DepEd memo. It is the supervisor's decision or instructions to adopt it. Oh, so talagang susundin daw.
Very well said.
Okay, the lesson log. Okay, so let's take another question here. This is interesting. If a student failed in bio grade 7, is it a prerequisite for bio grade 8? Or it does not matter as long as a student passes science in grade 7? Yes, any comments from our presenters?
Ma'am Sel, last time I checked the guidelines for assessment from DepEd, I didn't see any descriptive marking system there that is labeled failing. I mean, is the lowest possible mark that the student can get per quarter is B, that's beginning or beginner. Is that failing?
Okay.
I find it very logical to start, for example, the school year with B, and then later on, should there be improvement on the performance of the student, the student will get a mark of P, that's for proficient, and the like. I'm not sure, but I think it's logical.
I don't know exactly how a student who fails just one quarter, if at all, na fail nga siya, no, sa quarter na yon for science, because grade 7 science is now divided into 4 quarters, and each quarter is allotted one discipline or science area. So, hindi siguro yung computation noon ay nakapako doon sa content area, kundi sa buong science subject for the entire year, no? So, I don't see masyadong komplikado yata yung hindi siya pwedeng pumunta doon sa susunod na science, na biology sa susunod na grade level. Oo, hindi yata ganon. Oo. No? So it's a science taken as one entire subject. It just happens. Oh, yeah, hindi siguro ganon ang interpretation dapat dyan. Okay. Um, well, let me take this opportunity to thank Ma'am Jean Luyon for sharing with us the information about DepEd Order number 70. I think it will all benefit us if we take a look at this latest DepEd Order in relation to lesson planning in the K-12 curriculum. Okay, again, here's another question. If some of our students got B and D, is it okay if we do the remedial during the, what is CL? Collaborative learning time? Collaborative or independent learning time? Again, if I remember correctly, yung sinagot po sa atin ni Dr. Tan dito, it's really the school's decision, and if that is also a division level decision, how to make use of these extra hours during the week can be used for remedial purposes, can be used for really student collaborative or independent work. No? So again, hindi po sa aming manggagaling yung sagot. No? The school will have to determine when to use and how to use this extra learning time for students. Would you like
There's a question
to
here from the Dasmariñas science teachers. The one on, is it possible if we can conduct the activities for 2 days?
Yes.
Can we have
I think that question came immediately after Module 2. So that must be one of those activities in Module 2. 2 days. So
2 days?
For the
2 days, which would mean
is
2
it
hours?
possible we conduct the activities for 2 days? Does this mean all the activities in the modules for 2 days? 2 days means 2 hours in the science
Yeah, that's what I would like to know.
time of 1 hour a day. 2 days means 2 hours.
Science. Can we hear from the Dasmariñas science teachers, please?
Yes, that's from the Dasmariñas science teachers. Maybe we can answer them. They say choppy tayo dun sa kanila sa Dasmariñas science teachers group. Maybe we can use the chat window to type in the answers. They're asking for the activities to be covered in 2 days. What do you say about that? Let's see. Let me post that content. Po natin dito kung alin dito yung sinasabi po nila. I'm changing now the presentation to the module 2 presentation. So if
Yes.
the Dasmariñas folks can type in the chat pod, ano po yung activities na sinasabi ninyo na pwede bang tapusin ng 2 days? Paki-type lang po. So if we review the activities in module 2, sige nga, Dulce, we can look at the activity in module 2 and see which ones exceed 2 days or 2 hours. Yung sa microscope,
Activity
okay.
1.
Yun daw sa microscope.
Would
There are
definitely
two
be-
sets.
Well, there are two sets.
Yeah, one
let
is
me see.
the preliminary activity,
Microscope.
and the other one is activity 2.
Okay. Yeah, it's possible. As I was saying then, depending on the speed of the students, some teachers have told me that their fast learners could finish this in 2 days. That would mean 2 meetings, no? That's 2 hours. It is it has something to do with the number of microscopes that they have. Kindly kindly direct your attention to the answer. Kasi po kaming microscope, ma'am, iisa lang po ang nagana sa amin.
They just have one functional microscope, and they
One
have
functional microscope.
90 students.
Yeah, and they said they have 90 students. Or even 97.
Isa lang ang gumaganang microscope.
Yes. So that will
Yeah, I
really
think
take
their
time.
issue is
If they require students physical facilities.
So what can teachers or what can schools without computers or almost no microscopes do with module 2?
Microscope.
So more or less they just perform the first activity.
The first
Ma'am,
activity.
Ma'am Dulce,
So matatapos niyo yung module
Yes,
module
is the
2 in
activity on
Yes, Mike,
is the
are you
activity on the
saying
use of
something,
light
Mike?
microscope compulsory, Ma'am Dulce?
Yes.
What I'm saying, Ma'am Cel, is yeah, this is for Ma'am Dulce, Ma'am Cel. Ma'am Dulce, is the activity of the use of the light microscope compulsory?
No, it's optional actually.
Yeah, okay. So I think that
Yeah,
settles
so
it.
that's supposed to answer the question. Yeah.
Well, there is another option. One teacher was telling me then that that procedure A, the microscope, its parts and their functions, they do this as a demonstration if they have only one microscope.
Okay.
They can do
Okay.
a demonstration
So-
of procedure A. But they definitely what they do, some teachers would just demonstrate also B and C and just let the students look through after the teacher has prepared the wet mount.
Ah,
The
okay.
same with
So,
procedures.
let me type that in. Procedures A and B are done as demos. Then students take turns viewing the specimens in procedure C. Tama ba? Is my note correct? For the use of the light microscope, Dulce was saying that procedures A, where the students identify the parts and functions of the microscope and then make a wet mount. This is a teacher-led demonstration if there's only one microscope in the school for the class. And then as the teacher completes setting up the wet mount for observing the specimens, the students take turns in looking at the microscope. Tama po ba yon?
In observing or looking at the specimen.
Yes. So, ganun po yung setup nung
Yes, they take turns.
light microscope activity. So, which
Yeah.
means again that if you complete the activity procedures A and B in one meeting, because it's a demo anyway, and then get the students to observe the specimens in the second period, on the second day, definitely, I think you can finish this light microscope activity in just 2 days.
2 days.
No?
Right.
Di ba? Kasi 2 to 3 hours naman yan. Oo. So, yung demo, kasi wala namang magagawa. Talagang demo lang. Dahil iisa lang yung inyong microscope.
Yes. Yes.
And as
I
Mike has
also,
mentioned earlier,
suggested
optional
here-
din naman talaga ito.
Yeah. Yeah. But I answered them in the chat space area
Mhm.
through email the details
Okay.
of the problems with the other microscopes, because they said they only have one functional, which means they have others which are either, I don't know what happened to the others, but I suggested to them that they email us the problems with the other microscopes so that we can take a look perhaps, and suggested that we have a material here which they can have, okay, in our bookstore, which details on how to take care of microscopes and then make simple maintenance or troubleshooting of some, what do you call this, problems with the microscope. And they said, okay, ma'am.
Okay. Okay. So, there are at least two more questions, last two questions here in our list. One says that, this is from Ma'am Jean, no? Our problem is, what if we can't finish the whole modules because of a lot of interventions? I think this means this is intervening activities, no? Hindi nila matapos. Tapusin nyo, hanapan nyo gawan nyo ng paraan. Any note on that, Mike?
Yes, Ma'am Sal. When we wrote the modules and the teaching guides, we actually reserved rooms for a lot of these intervening events, such that should they be confronted with a lot of these intervening events, they would still have time to make the necessary adjustments and finish the necessary activities.
So, again, this is unsolicited advice, but I would still think that we cannot, since these are intervening events anyway, this really are things we don't want to happen, but have no control over. But then again, pagka mas marami yung intervening activities, the more reason we really need to plan or strategize our class time allotment, no? So, kailangan yata planuhin kung paano ngayon ipapasok yung mga di pa natatapos na modules. Otherwise, the intervening activities don't end throughout the year. Ang dami yan, maraming beses. So, kung palagi na lang wala tayong gagawing paraan, each time na may intervening activity, ang mangyayari lang sa atin, isang module sa isang quarter, ay talagang kalugian na ng mga estudyante yon. No? If I were the teacher, then I really would like to do, to make sure that the modules that were not yet covered will have to be covered in one way or another. may ceiling grade po ba ang mga bata sa special science class sa bawat grading period para sila mapabilang sa list of students with honors? Kasi may naririnig po kami na pag below 87 ang grade ng bata, madi-disqualify na daw po siya. I think kayo po, no, sa school ninyo, I think there are existing implementing guidelines with regards assigning students in a special science class. We don't know exactly. I don't think we have information about the ceiling grade for students in special science classes. Folks, those of you, our teachers here, attendees here, who also have special science classes in their schools, would you like to type in answer to this question? Do you have any idea about the grade, the minimum grade that students need to maintain in order to stay at the special science class? Any idea on that? I don't think we have, no? Kami sa NISMED, hindi yata kami familiar sa ganito. And maybe you can ask, yun pong nagsabi na may narinig po kami, nakasulat dito, no? May naririnig po kasi kami na pag below 87 ang grade ng bata, madi-disqualify na daw po siya. I think you should verify, no? Sino yung nagsabi noon? So, tanungin nyo na din siguro sila, no? About that. There's a question, what is the English term of LUMAT? I think Dr. Risa has answered this earlier in the chat pod.
Yeah.
Again, from the Marina science teacher, so ma'am, can we extend the suggested number of days? Posible po ba yun? Posible siguro, no? Basta meron kayong ganung araw. If you can find time, everything is possible. Parang hirap naman sabihin na, no, you can't. No? Or yes, you should. No? You would know exactly what you've missed and how much this is going to mean in terms of losses in learning sa mga bata. I think may kaya naman ng school ninyo, no, mag-make ng decision about extending number of days. More questions? Oo. I think you should strategize, no? Yung mga hindi nako-cover gawa nung may mga intervening events at saka
bagyo,
remedial requirements. I think you really have to strategize within the school about how to deal with that. Okay, so well, there's a clarification here from Congressional National High. Ang concern daw nila ay nasa ranking. So again, may I just repeat, we don't have information about this to be able to answer you meaningfully.
Baka hindi na.
I think we have not skipped any question. Okay, so I think we've covered the questions and we're ready for Mike to wrap up and the session. Mike, would you offer, would you like
thank you so much.
to show a wrap up, or we continue on with the closing activity already? There's another one here about the units. Yeah. How many units per subject? I don't think we have an idea about that. May units ba sa subject?
No idea pa.
Number of units ng bawat subjects. Number of units ng bawat subjects para makuha namin yung average grade. I don't think we have this information, no? This detailed information. Number of units sa bawat subject.
Yeah.
I don't think this is within our knowledge.
Yeah.
Okay, again, here is an input. Yung po lahat sa grading system, Dr. Tan has informed us that DepEd is going to release soon. So, dapat nga darating na yun, no? Kasi ang dami ng mga tanong ng mga teachers about the grading system. And I hope that will really be addressed soon, no? Maraming concerns about the grading system. So, kasama na siguro yung maraming tanong from our friends, the teacher friends from the division of Dasmariñas and also the Congressional National High School, no? Hintayin natin, bantayan natin, o email natin sila o text ba natin sila para ilabas na nila kaagad yung more detailed and revised grading system. Okay, so let me switch over to our again, we would like to really thank you, those of you who have stayed on with us for this second webinar in our series. We would like you to share with us your feedback about today's webinar, and we're sharing with you the links here. So I will click now this survey form so that it will appear on your screens. In case you have pop-up blockers, please disable them when you see one. So right now I'm pushing on all of your screens this survey form, so kindly find time. This is very short, you can quickly finish it. We would really appreciate your feedback.
Ma'am Sel, while they do this, do I do the wrap-up now?
Yes, Mike, please.
Okay, Ma'am Sel, thank you. To wrap up, the topics for Q2, the second quarter, are sequenced along a narrative, a storyline that we think makes sense from a grade 7 learner's experience of the world. The activities serve as their guide in navigating the concepts, principles, and processes of biology, and how they all provide a landscape of science where biology is but a part. To the bio majors, we hope you'll find Q2, the second quarter, not as bio-light, but as bio just right. And to the non-bio majors, we are here to help. Please do keep in touch. Again, thank you, dear participants. We hope to see you all again in the next webinar. God bless us all.
Okay, I'm sorry, Dasmariñas science teachers, I missed this. If the grade is B, do we need to indicate the red color in the student's grade? What is the practice in is there a color code for B, for approaching proficiency and so on? Oh, I think you have to make decisions with regards to this on the basis of what is the practice and what guideline do you have in implementing the use of color codes for student grades, no? Mas po yatang kayo yung makakasagot dito sa tanong na to. So again, you can always ask your school, your principal or your division superintendents, and the other school governing council in your respective areas. you may, I think, mas masasagot kayo ng inyong mga co-teachers or principals or heads, school heads dito, no, sa tanong na to. Okay, so as a final announcement, I would like to inform everyone that the PowerPoint presentations that you've seen today will be posted in the discussion thread in KaSaMa Teachers. Also, this webinar is being recorded, and a link to the recording again will be posted in KaSaMa Teachers, along with the questions that you have raised during the webinar today. Again, thank you for attending today's webinar. Join us again on September 28th for the third in the series. And our topic will be the focus for the third grading period or the third quarter, which is force, motion, and energy. Maraming salamat po, and please indicate on the chat pad if you have answered the survey. Pakisagot po para po magkaroon kami ng data about your feedback. Thank you very much, everyone. Thank you, Dr. Risa, Mike, and Dulce for taking time to present to us the second quarter topic. Okay, see you all again next month.